Good morning, everyone. Welcome to our sixth episode of the webinar series of Ecogen. And the theme today is actually in a two-part, and it's called Innovations Beyond Traditional Breeding. And we'll hear about different tools that has been used recently or in the last few years to improve breeding systems. So before starting, I would like to introduce our different speakers today. We will have Adrián from and Terhi, Birgit, Luca, and Velma. And I'm very grateful for the five of you to be here today and to make this webinar a success. And we are very glad to hear about the different tools that you developed within your team. But before starting, I would like to go over the agenda. So we're starting now with the introduction, and then we'll have Terhi from HoloRuminant, and then we'll have Velma, who's also from HoloRuminant. And afterwards, we'll have Adrián from Rumigen, and Birgit and Luca from Livestock. They will all have 12 minutes to present, and we'll have the opportunity to have an open discussion at the end where the room will be open for you to ask questions, and if you're shy, you can ask a question in the chat. I would like to go through a few house rules, so please mute yourself when you're not speaking to avoid background noise and to not disturb the speakers during the presentation and the Q&A. If you have a question, please add it in the comment when it's during the presentation of the speakers. Raise your hand when you want to speak, and we will give you the floor. And please introduce yourself and add before your questions. And there's a recording right now, so we would be grateful, like, if you don't want to be in the recording because it will be on YouTube for people to watch again, please keep your camera off. And if there's any technical issue, just write it in the chat. And also you can send us an email on projects@efab.info. So thank you. And I have a few words about our roles. So Ecogen is a collaborative research cluster. It is led by HoloRuminant, and it's a partnership with Real Livestock, 3D Omics, Rumigen, and Geronimo. So in the last 5 webinar series, you would have heard different sessions by the different projects where they talk about what they're doing and how those projects can be linked together. So the idea is to explore innovative breeding strategies on microbiome research and advanced technology, among others. So the focus areas include developing sustainable practices, genetics research, using different technologies that you will also hear today, investigating microbiomes, holistic approach, but of course, system reengineering. So really designing resilient and sustainable livestock systems. So I'm Gina Kardik, and I'm from EFAB. EFAB is a Brussels-based company where we have members from the breeding sector, but we also have research and institute from an academia, and this together they form part of the FabreTP, which is a technological platform for the farm animal breeding and reproduction. Some of you might be members and some of you might not, but it's just an introduction to show what we do. So what do? We actually engage in dialogue with policymakers and stakeholders. We support and promote responsible animal breeding through Code FABAR. And also we promote and support research innovation through our role for different projects, including the 3 projects mentioned today, Geronimo, Rumigen, and HoloRuminant. And we also advocate for the proportionate and science-based policy and legislation, which means that we try to bridge the gap between science and policymaking to make sure that policymakers takes the right decision based on science. We are also partners with different organizations, including Animal Task Force, Aquaculture Advisory Council, because as you may know, we represent all species, including aquaculture and insects. So if you have any question about anything regarding EFAB, you can always come to me, Chala, or Maureen, who are in the room. But if you want to know more about the different projects that's in Ecogen, please get in touch, and you can scan the QR code to be able to have access to all those 5 projects. So I will stop sharing now, and I would like to welcome our first
Gina, you're muted. Can you unmute yourself?
Did you hear everything else though?
Yeah, everything was fine. Yeah.
Okay. So I would like to introduce Terhi. Terhi is a senior scientist at the Natural Resource Institute Finland, Luca. Her work explores how cells model can replace invasive method to study genetic resistance in dairy cattle, helping us link genetics to animal health and welfare. Thank you, Terhi, for being here today. I'll give you the floor.
Thank you, and good morning, everybody. So first, I'd like to thank the webinar organizers for giving me this opportunity to talk here. And I will really tell you about the cell models and how we have been using them to study genetic resistance of mastitis. So I will really briefly first tell you about the cell models and their usage in animal research. And then I talk about mastitis and how we have tried to identify genomic features that are underlying the mastitis resistance. And for that, we use the cell models. So according to literature, the first animal cell model, or actually it was the animal tissue model, was created already in 1910 by Ross Harrison. The model was so-called hanging drop culture from the peripheral nervous system of frogs. That model was followed with different tissue culturing models, but as explanted tissue cultures had their problems, the next step was to use primary cell cultures that consist of single cell types and allow to study the behavior of specific cells. Primary cells are good, but they are rather limited. They have rather limited lifespan, and as they are collected from various animals, they have also varying genotypes. To overcome those issues, immortal cell lines were started to use. Cell lines allow studies to be repeated in controlled conditions, but they tend to change their phenotype and genotypes over time. Nowadays, also 3D cultures, organoids, and organs on a chip are used. Those contain multiple cell types and are mimicking more closely in vivo conditions that are single cell cultures doing. However, they are quite difficult and costly to establish, and that is really limiting the usage of the 3D and so on models. So for what animal cell models can be used? Here are just some examples, but they have been used and are used, for example, for developmental biology studies, toxicology and pharmacological studies, for functional validation of candidate regulatory variants, and to see, for example, the gene functions in response to pathogens. So, next, I will introduce to you how we used milk-derived bovine primary epithelial mammary cells to study mastitis resistance. This work is published in these 2 publications, so feel free to look more if something is left unclear after this. Our aim was to use the non-invasive approach and in vitro to identify genomic features that are really behind the mastitis resistance in dairy cattle. We wanted especially to study cows' initial cellular response differences when they are facing Escherichia coli in the mammary gland. And as you all mostly are aware, mastitis is the inflammation of the mammary gland, and it's typically caused by bacterial infection. Mastitis is a huge economical and animal welfare issue, but it's also the most common cause of antibiotic usage in dairy cattle. This is, of course, creating the possibility for pathogens to become resistant to antimicrobials. Mastitis has been studied really intensively, but still better understanding of biological background is needed, really to improve the breeding and the management of the mastitis. What we did was that we collected milk from Nordic red cows with either having the high or low genomic index value for udder health. Milk donor animals were all also whole genome sequenced. And then we extracted the primary epithelial cells from the milk by centrifugation, cultured them, and challenged cells with the E. coli. The motivation to use the epithelial cells is that they really contribute to the innate immune system of the bovine mammary gland. And the E. coli strain we used was isolated from the Nordic red cow having mastitis. E. coli is one of the major mastitis pathogens causing acute clinical mastitis, and it often causes the really severe symptoms. And that's the reason that it's the major concern in dairy cattle. After we challenged the cells, we harvested them, extracted the total RNA, and sequenced that. That was done for both challenged and unchallenged cells. When we had transcriptome sequences in our hands, we looked for differentially expressed genes. We did the co-enrichment analysis and cake pathway enrichment analysis. And we also looked for overlaps with the known GWAS results from the Nordic red population. We also did EQTL analysis, even though we are fully aware that our study population was rather small for that. So, as a first results, we were really happy to see that the extraction, culturing, and cryopreservation of these mammary epithelial cells that were extracted from the milk was successful. Also, what we see was that the gene expression differences we see the differences in the gene expression between the challenge and control cells, and we saw upregulation of immunopathways and identified also immune-related candidate genes. So, firstly, we could show that epithelial cells that were extracted from the milk are actually serving as a quite handy tool to measure innate immune responses against pathogens. So it can be used for whatever pathogens, we just happen to use the E. coli here. When we looked for the differences of the gene expression between cells derived from the cows having high or low genomic index value for other health, we saw that there was markedly higher number of differentially expressed genes in the low mastitis resistant group after challenge compared to the high mastitis resistant group. Also, based on the co-analysis, we saw that adaptive immune responses terms appeared enriched in the low mastitis resistant group later than in the high mastitis resistant group. Gene pathway analysis was also further supporting that, and it revealed that high mastitis resistant group shows earlier innate immune activity, while the low mastitis resistant group shifts towards adaptive immune responses and later in the challenge. When we investigated the overlaps between the no mastitis QTL, two regions from chromosome 6 and 9 were having differentially expressed gene enriched. Chromosome 6 region is a really well-known mastitis region, mastitis QTL, and it has also the candidate gene GC identified. In our study, that region was both enriched with the differentially expressed genes, but also had an EQTL, so further just supporting the findings that have been done earlier. So now, all ready to conclude. The low mastitis genetic resistant group showed slightly delayed adaptive immune responses and early metabolic changes. This may indicate the possible underlying genetic factors that are influencing the transcriptomic reactivity. We also found that the key immune-related genes that are identified already before responded in the both groups, but we didn't see any group-specific differences at early infection stages. And finally, we did not see too many overlaps between differentially expressed genes to KIS EQTLs identified in this study and GWAS identified SNPs from the previous studies. That may indicate that the early epithelial immune response genes may not drive phenotypic mastitis resistance. And finally, I'd like to show to you our team that have members from 3 different institutes. So Natural Resources Institute Finland, or Luca, University of Helsinki and Aarhus University. And our work was funded by Research Council of Finland and the EU project HoloRuminant. So thank you for listening, and yeah, good.
Thank you very much, Terhi. So you get some applause, I can see. I would say if you have questions right now, please put it in the chat, else we'll keep all the questions at the end of the session. Thank you very much, Terhi. It was really interesting. We'll now pass the floor to your colleague, who's also in HoloRuminant, and it's Velma. So Velma is a researcher at the Norwegian University, so NMBU. She studies ruminants, HoloRomics, and using multi-omic tool to better understand how microbial communities in the rumen influence animal health and productivity. Velma, I give you the floor, and we'll keep the question for the end. Thank you.
Yes, thank you so much, Gina, and thank you for the opportunity to present our work in this session, even though I would say we are indeed quite far from breeding, which is a keyword in the title, but I would like to try to convince you that we also need to think about microbes in whatever context that we work when we are looking at ruminants. In the very title of the HoloRuminant project, which is my main project at the moment, we have this holo and the tagline understanding microbiomes of the ruminant holobiont. So as this is also in my title of the talk today, I would first like to tell you what actually we mean by this. So to start with, what is a holobiont? That is a sort of philosophy of considering a larger host organism, such as an animal. It could even be a plant, but now we're talking in the animal world, and the microbiomes of that animal. So all the microbes that are living in and on that larger animal, and thinking of them as one single unit that is working together. In the case of ruminants, they are a very good example of a holobiont because really they could not function without their microbes. They are completely dependent on microbes that are able to digest the plant matter that these animals are eating. The animal itself would not be able to do that. So that is a holobiont. What is then holo-omics? That is basically just using some kinds of approaches or methods where these organisms are studied as a combined unit. So holo-omics is a research philosophy of looking at holobionts. Very often how we do holo-omics is to use multi-omics, another important word here, which basically just means using a lot of different methods together to look at different molecular layers that we can measure in these organisms. So here we have in our diagram, sort of on the horizontal axis, the holo-omics, so the host and the microbiome, which are then considered together. And then on the vertical, we have multi-omics, the different methods, starting from the genome. People also sometimes say hologenomics to look at the same thing, but we like to say holo-omics to sound more comprehensive. After the genome, we can look at the metatranscriptome and the transcriptome for the RNA level. Then we can look at the proteins with proteomics or metaproteomics. And then finally, at the end of the workflow, sort of are the metabolites, so all the small molecules that are getting produced at the end. And I believe we will also have another talk about metabolomics today. So that is the sort of philosophy, the background that we operate with. And what we then did in the study that I want to look at today was to apply these tools to a study group of, well, in the end, 24 animals. But we started with 40 plus 40 beef cattle of different breeds. Initially, this was meant to be a study related to methane mitigation, where the animals would be given red seaweed that takes their methane levels down. But we got a bad batch of seaweed that we couldn't use. And in the end, this just then pivoted into a study where we followed these animals in very controlled conditions for many weeks, measured a number of key performance traits. And at the end, we measured their methane levels in respiration chambers because methane was of interest here. For the full set of 80 animals, we performed your sort of classical rumen microbiome analysis using the 16S marker gene, which is kind of the general gold standard that we do just to see which microbes are there and how many of them you have in relation to other microbes. And then for the selected 24 animals, that represented equal amounts of both breeds and different methane levels, we did a very comprehensive characterization of not just the rumen contents, but also liver samples and rumen epithelial samples to get more into the host side of things. And we did all of these different molecular measurements that I was talking about, so the genome, transcriptome, proteome, and some metabolomics for these different samples that we had collected. Now, where this study ended up going was also sort of serendipitous, because when we started looking at the microbiome with this basic 16S marker gene analysis, which is just showing us now bacteria and archaea that are present in the rumen, we found a pattern that was really intriguing to us. So here is a very common type of plot that we like to do in microbiome analysis, where we just condense all the variation on 2 axes. Each point here reflects the microbial community of one animal, and the closer the points are, the more similar those animals' rumen microbes are. And it is really, at least to me, very evident here that the points, so the microbial communities, seem to form 2 clusters, 2 different clouds that are quite separate from each other. You could kind of draw a line in this figure to split those apart. And first we thought, okay, maybe this is the breed, but it wasn't the breed that is different. It wasn't the methane. We got worried that it's something technical, but we checked all of our technical handling batches and animal housing and everything we could think of. Nothing was explaining this. So in the end, we thought, okay, maybe this is something actually biologically interesting that we need to investigate further. We ended up naming these 2 kinds of microbial communities as rumen community types A and B for historical reasons that I will very soon get back to. But first, I would like to tell you that this was not just a feature of the 16S marker gene microbial community analysis, but when we started to look at all of these other omics that we had measured, the metatranscriptomics, metaproteomics, metabolomics, we were seeing this same community structure reflected in all of these different layers. So specifically here, we see a very big difference in digesta and wall metatranscriptome, digesta metaproteome, and some differences in the metabolomics. I won't go into the technical details of what we are actually looking at here, but just you have to believe me that we saw a very strong difference between these different community types in almost all the omics that we looked at, which was very intriguing. So of course, the next question is, what are these differences then? Are there specific groups of organisms that are different between these communities? Of course there will be, because the communities are based on amounts of different microbes. So this is one way that we looked at this. And what really was a bit surprising and really cool to us was, instead of just seeing differences in bacteria and archaea, which are generally the microbes that people know best, we also saw a lot of very strong differences in protozoa, or ciliates, which are some of the most important eukaryotes in the rumen. They are quite big and quite numerous, but they are still kind of understudied, I would say, because they are challenging to look at, aside from using very traditional microscopy approaches. They have very complicated genomes, so the genetic-based tools to study them are very kind of unexplored so far. We saw, for example, a lot of differences in having a lot of genus Epidinium ciliates in the type B animals, and a lot of Entodinium isotricha and polyplastron in the type A animals. Now, I mentioned that there is a historical reason for the A and B, and indeed, when we looked into the literature, as we were not ciliate researchers, we were quite excited to find that this was actually a known thing that had been reported way back in the 60s. We rarely get to cite 60s when we do the modern omics kind of studies. They had already figured out, based on microscopy, that it seems that animals often have either type A or type B ciliate populations, and the markers, the most common ciliate species, were some of the same ones that we saw. But we didn't entirely just reinvent the wheel here, because this was just looking at the amounts of these specific ciliate species, but we had first seen a difference in the bacteria and archaea without even thinking about ciliates. And we saw this on all of these functional measurements of transcripts and proteins that we were looking at. So clearly this had a sort of more fundamental difference going on than the initial studies were thinking about for the ciliates. And to go on to the more sort of holo-omic angles, we also wanted to look at the host. Is there actually an influence of these different community types all the way to the animal itself? Well, looking at the more animal origin omics from the rumen wall or the liver, there were not as strong differences there. There were some indications or trends of differences. Of course, this is quite a small data set as well, but potentially maybe something going on there. What we thought also really important for the host was that we had these metabolite differences. For example, in volatile fatty acids, which are extremely important energy sources for the host that are produced by the microbes, we had a lot more butyrate in the type B animals and a lot more propionate in the type A animals. So there also an important difference. And we also had, for example, higher levels of some amino acids in the A-type animals. So this kind of difference that we see at the sort of downstream end of our omics that we looked at could be very important and influential for the host functioning. And finally, to mention, all of these previous slides and figures were from our published study that came out this summer. You have the citation details at the bottom of the slides. This figure was not in the study because we didn't really go in depth with these kinds of questions yet. But I thought it's very interesting for the present session. Initially, I said we didn't see differences in breed or differences in rumen when we were trying to explain this split into two clusters that we saw. But actually, the story is a little bit more complicated than that. So if we look at methane output just for rumen community type A versus B, we don't see a difference. But if we also divide the animals by breed, because we had two different breeds, then we see a difference specifically in the Luing animals, which is a Scottish traditional breed. So there the type B rumen community seems to come with a higher methane output, but not in the Aberdeen Angus cross animals. So clearly there is some kind of interaction, cross activity going on between the animal breeds. So maybe the genetic background and the rumen community type as for when it comes to methane output. And that is very cool and something that I think is worth looking into further that we did not do yet, but hopefully will at some point. So to wrap this up, we found these two different rumen microbial community types in animals. Even though the animals were in identical experimental conditions, they still developed these different community types, and they were reflected in almost all the molecular layers that we measured. We think rumen ciliates are the most important driver behind the community differences, and this is very closely resembling these traditional microscopy-based categories, but seems to be much more far-reaching than was thought initially. The effects on the host are not quite as strong as on the microbial community, but definitely they warrant further study. And this could really have implications far beyond research, for example, for breeding, for diet optimization, because the different communities are digesting diets in different ways, and of course the potential for microbiome-based interventions, which would be very interesting for methane mitigation, for example. And I would just like to wrap up by saying that to us, full understanding of an animal really requires understanding the contribution of its microbial community. You can only really get the whole picture if you look at them together. And of course, I need to acknowledge quite a few people as well for such a big study. And most of all, Phil, who is our work package leader in HoloRuminant, and Carl, who was a PhD student, graduated last January and did a lot of the work that I've been showing you today. Thank you.
Thank you very much. Excellent timing. So thank you again for this deep dive into the rumen microbiome and how the hosts and microbes function together as one system. I think it's really interesting to go beyond, to go within the animal to better understand its function. So now we'll shift from the microbial ecosystem to the molecular layer that connects environment and genetics. So in a nutshell, we're going to talk about epigenetics. So our next speaker is Adrián from India, and he's from the Rumigen project, and he will show how epigenetic information can take breeding beyond genetics. And it shows how the environment leaves molecular markers on the genome of cattle. Thank you, Adrián.
Thank you very much for the introduction and for the opportunity of being here today. And also I would like to extend my gratitude to the EFAB team for organizing this seminar series. So recently we've started talking about going beyond genetics. I've quite a lot of used this in my presentation, but I have seen other people saying that. And I think it's because we are starting to realize that it's not only genetics what we have to look into. So we know that our DNA has two layers of information. all of you in the animal breeding sector are quite familiar with genetics. We know it's this layer is resistant to change. It's the one we inherit from our parents, and it's the one we are targeting right now, the one we select our animals for. But we also have the layer of epigenetics. It's a more dynamic layer and help us to adapt to our environment. So imagine we have a heat wave and we need ourselves or the animals to swiftly adapt to this change in the temperature. So that's probably epigenetic regulating what's going on with us. But there's a lot of flavors to this layer. There are a lot of different epigenetic marks. We have prions, we have non-coding RNAs, we have chromatin condensation, histone modification, and the one we are focusing on today is methylation. So methylation marks consist of, it's simply the addition that happens naturally of a methyl group to the fifth carbon of a cytosine. So we have our cytosines and they get transformed into a 5-methyl cytosine or 5-MC. They are usually associated to the establishment and maintenance of gene expression, and it's a bit of an oversimplification of the reality, but if we looked into the promoter regions, when we have an unmethylated CpG or island in that region, it's usually associated with gene expression. But if we have a methylated promoter, which will be the case on the lower image on the right-hand side of your screen, it's usually associated with a gene repression because the transcription factors cannot bind to the promoter region. And even if the genetic potential is there, if the genes we selected for are there, we are not going to be able to see the phenotype because of this epigenetic layer. And of course, this is an oversimplification of the reality, and there are a lot of exceptions to what I just said. Until now, there were two major ways of detecting these methylation marks. We had bisulfite sequencing that consists of the addition of a chemical, a bisulfite, that transforms methylated cytosines into uracils, while methylated cytosines remain the same. And then using two different cycles of PCRs, we can detect the cytosine that changed and those that didn't, and understand which one were methylated and which one weren't. But in our lab, we use an opposed sequencing that is based on the disruption of electrical current. So when a DNA strand passes through the pore, each one of the nucleotides had a different electric profile that we can capture and understand what's going through the pore. And the cool thing here is that methylated cytosines and unmethylated cytosine have different profile that we can detect. But these techniques are quite, they are not cheap. They are quite hard to apply at commercial level. And that's why in the RumiGen project, the epigenetic work packages 6 and 7, there has been a huge international effort that have been led by the INRA, Ellen Kiefer and Valentin Costas. I'm sorry, I butcher your French pronunciation of your name probably. But we have developed an epigenetic, an epigenotyping array that has more like 45,000 CpGs associated with different traits of interest. So we have traits, CpGs covering the traits of parity, Staphylococcus aureus, as is a microorganism associated to mastitis, embryo development, fertility, even breed, age. So a lot of CpGs associated to different traits that we are starting to explore right now. One of the main reasons we did that was because it's easier to apply this tool in commercial farms. So for example, we work with this commercial farm in northern Spain. We have been doing all the analysis I will show with them. They have over 1,000 lactating cows, but we also need to be able to get non-invasive tissues. So this EpiChip is also designed to be able to use blood samples to detect the methylation in this tissue. So we can go to the farms and say, I just need to take a blood sample of your animal, and that's easier than say I need a liver sample or a different kind of tissue. In our lab in 2012, we started analyzing differences between those cows that were gestated by lactating cows and those that were gestated by non-lactating cows. Because in this work, our PI, Oscar Gonzalez-Recio, and some collaborators, they detected that the more lactations the mother of the cow undercame during her life, the worse milk production the cows will have during their adulthood. So as you can see, in first, second, and third lactation, we see decrease in the milk yield of the cow when compared to those that were gestated by non-lactating cows. They hypothesized that it was probably due to metabolic stress, because during the pregnancy in dairy cattle, there is a period of time where lactation and gestation overlap. So the more the energy requirements for meal production cannot be only met by the dry matter intake, and this leads to a decrease in the body weight. It does not matter how much the cow eats, the energy cannot get there. So we believe that there is a metabolic stress that is impacting the oocyte. The thing is that these do not happen in primiparous cows. So cows that are not lactating will not have this problem when they are gestating a calf. So we believe that there are some epigenetic differences there. So probably this difference in the milk yield will be explained by epigenetics, but back in 2012 they could not prove that, and it was only an hypothesis. So this is what we have been testing with the ruminant chip. For that, we have blood samples coming from 466 calves that have been both genotyped and epigenotyped. We have used bioinformatic pipeline that I'm really not going to get into, and apply some statistical models to integrate all these sources of information, the phenotype of the mother, the genotypes, and the epigenetic information. Using this Bayesian approach, we can get heritability estimates of the methylation of every CpG, and we have seen that the heritability range from, it's quite concentrated around 0.2, but some of the CpGs have higher heritabilities, and we are looking into them with more detail because they have heritability of around 0.7, which goes in line with the studies from other groups. It's true that we have only used one head, so probably this could be an overestimation, and I would like to highlight that methylation itself is not inherited. What we inherit is the genetic control of the methylation, because methylation is reaching in every generation. There is a big erosion of the marks during the pregnancy and the embryo development. Yeah, we also look at different genetic features to see if the heritability in the marks was different depending on where it was located in the genome, but it did not matter. So the heritability is ranged again around 0.2. Using this Bayesian approach, we can also see until what extent the maternal phenotype is affecting the methylation, and we have seen that at genome-wide level, solely uniquely the maternal lactation could explain only around 1% of the variance in the methylation of each CpG. And what I'm going to show you now, it will look like a Manhattan plot, but it's not a Manhattan plot. So what you have on the y-axis is the percentage of variance that on the methylation frequency of each CpG that is only explained by the maternal phenotype. So we have removed every other factor and focused only on the maternal effect on these CpGs. And we have found some CpGs that whose variation is explained by more than 10% by the maternal lactation. So what we did was only adding the genetic of the animal, but the lactation status of the mother, and this 10% threshold was from the milk chosen, but we believe that is a quite strong threshold. And we have found some interesting genes. For example, we have found ADGRL3, that is a gene that is regulates the neurogenesis of the embryo development. So kind of strengthened our initial hypothesis. And around 26% of the variance in the methylation of the CpG was only explained by the maternal lactation. We have found also other genes that were associated with the regulation of adipogenesis and lipid synthesis, which can have an impact on milk production during the adulthood of the animals, and 24% of the variance of the CpG was explained by the maternal lactation status. So we know that there are some marks that are affected by the maternal lactation status. We know that there are epigenetic marks that can have an impact on the animal well-being and health and production status. But how can we include this information into breeding program? And this is the hard part. So until now, we know that the more information we include, the better measurements we're going to get and estimations we're going to get. What we do until now was measure and register phenotypes of interest and use these animals as the parents for the next generation, combining this with genomic information. So our idea was, could we improve the estimation and the ability of our programs, our software to estimate heritabilities, breeding values, if we included epigenetics? And for those of you that are not familiar with breeding schemes, what we do is we believe that a phenotype can be explained by the mean of the population for the phenotype, some fixed effects and some random effects, and obviously a residue. And so the more information you include here, the more accurate prediction you'll have. So we have for example, the pedigree blobs, that you get a vague description of what's going on. We are now in the point where we add pedigree and genotype information, and we get good estimate and good prediction of the value of the animals. And also trying to add epigenetics. So what idea is that we are going to get even a more accurate prediction. So we have decomposed this random animal effect into an epigenetic modulated breeding value and an additive residual breeding value, but I don't really want to get into very much detail here. You can find all the information in a paper we recently published, but what I would like to show you is that if you look in the three first rows, this could be the estimates for a traditional model, where if we increase the heritability of methylation marks and we are not accounting for them, the genetic variance for the traits trait will increase. So we are capturing methylation effects within the genomic effects, and we are getting probably biased heritability estimates for these traits. But using the model we propose, we can separate the genetic effect and the methylation and the epigenetic effects, and get totally different and unbiased estimate for the heritability of these different traits. So there's this problem that epigenetics is very tissue specific and also time specific. So the next question is, we know that we can include this information, but when do we need to measure the animals? Because this is what we are seeing. We have a population of animals that we have sequenced an epigenotype during different times of their life, from the moment they were born to the moment they had their first parturition. And we have found that there are a lot of CPGs that get more methylated during time, and some of them get demethylated over time. So they are probably associated, if they overlap with a promoter region, those that get more methylated with a decreased gene expression, and those that get unmethylated with an increased gene expression. So we need to be sure of the dynamic behind each one of the CPG before we include them on the breeding programs. This is a study we have recently started, but and we're going to explore how methylation dynamics are changing over time is to promote this gene expression or repression. Because if we, as I said at the beginning, if we select the animals for their genetic potential, but then that genetic potential is switched off because of methylation marks, it will not matter at the end of the day. So we have found that aging is related to this alteration of DNA methylation. And the cool thing is that an animal can be naturally old, but epigenetically down. So we need to develop these epigenetic clocks to see if there is a difference between the actual age of the animal and the epigenetic life of the animal, because that could be associated with a longer and an improved functional longevity of the animals. Yeah, so we could also target for animals that are aging better and target for epigenetically healthier animals for breeding. Yeah, and just a couple of take-home messages for you at the end is that this new tool we have developed within the Ruminant project allows for a larger scale epigenetyping of samples, which also reduce cost and make it more accessible for commercial farms. We have found a set of CPGs that are associated with metabolic stress, confirming our initial hypothesis using this Ruminant tool. We know that this information can now be included into traditional breeding programs, but it's true that the models we are using by now are quite complex, even though we are gaining accuracy in the estimates. So we are working towards more simple equations to account for methylation in larger schemes. And there's also this idea that we could develop a chip that combines an array that combines SNPs and CPG information, so we can both genotype and epigenotypes the animals at the same time, reducing even more the cost. And I'm showing you a lot of results, but I didn't want to get very technical, but there is still a lot of work to do. We need to be very, very sure of the molecular biology that is behind all these methylation changes before applying them in commercial farms. So with this, I would like to thank you all for your attention, and thanks all the collaborators in this specific project. Thank you very much.
Thank you a lot, Adrián. And it's really interesting to see how epigenetic information can really complement the traditional tools that we have and help us understand this relationship between environment and how it shapes and the performance of an animal. So now we'll move from the molecular level to go a bit more like at the population level application. So our next speaker is Birgit, and she's from Wageningen University and Research, and she's also working in livestock. And she will discuss how combining genomic and metagenomic data can support selection for a more feed efficient and low methane dairy cattle. Thank you, Birgit. Give you the floor.
Can you hear me?
Yes, you are, yeah.
You see the screen properly?
Give me a sec. Yes, perfect.
Perfect, great. Well, thank you very much for the invitation to be here with you today and tell you a little bit about our work in the breeding work package of livestock. I would like first to thank the courses that you can see on the first slide. That is Coralia Manzanilla and David Flostock from our group in Wageningen. But I would also like to thank Oscar Gonzalez Recio from the University of Edinburgh. So we are closely working together here on this topic in the livestock work package. So one of the main objectives in livestock is how breeding can contribute to climate change mitigation. And here we focus on the use for methane emission, and then ultimately how can we use this information to develop new breeding strategies to contribute to reduce greenhouse gas emissions. So how are we going to do this, or what is our work about? So one of the focuses that we work across country. So we try to combine data across country to increase our reference populations for genomic predictions. So first, we focus on bringing phenotypes together for methane emissions of different populations across country. So we focus more on the host genomic side. But in addition, we are also looking at the rumen microbiome. And it was really nice talk that Velma just gave and saw the connections to methane as well. Really nice to see. So also here in livestock, we are looking how we could use this information. So I've split my talk in two parts. So first, I will tell you a little bit what we can do combining data across countries, and then showing some first results about how we could use the rumen microbiome information in breeding. So first, starting with a multi-country genomic prediction of methane emissions in dairy cattle. So this is work that was mainly carried out by Coralia from our group. So we have been working together here with Spain and Poland, where methane phenotypes and genotypes of the animals were shared between the three countries. So Netherlands, Spain, and Poland. This is easily said than sometimes done, I have to say. So bringing data together from different countries is also connected with some challenges. So first, we had to ensure that our measurements are kind of as close as possible or as equal as possible. So we have been working quite a lot on the measurement standardization, the trait definition across countries, and then estimated the genetic parameters, because of course we need to know that genetic correlation between the traits across the countries, and then ultimately go for a genomic prediction and estimate the breeding values for the trait of interest. So the objective here was, to determine whether using data from other countries enhances the accuracy of genomic prediction or not. And we have looked at three different scenarios here. So the first one is where we assume a genetic correlation between the traits of 0, so not at all the same trait. The second one is when we use the actual estimated genetic correlation. And the third one, if we assume the trait is really the same, so a genetic correlation of 1. Just a little bit about the trait. So the trait recorded in the different countries was methane concentration, recorded by different sniffer devices. You can see the pictures here. And on the right-hand side of the slide, you can see the data preparation that Coralia carried out. I'm not going into the detail here, so if you're interested in that one, you can see it on the slides, or we can discuss afterwards. A little bit more info about the actual data that was then available. So you can see in total there were 18,600 cows with methane phenotypes available. You can see here the different countries, Netherlands, Spain, and Poland. So the first were the number of weekly records included, then the number of cows per country, the number of herds, and the years the recording took place. So this was really a great data resource. I think it's actually the largest combined data set that is available at the moment for methane emission. And yeah, we were very curious about the results when combining the data. So this is the model that has been used. So it was a bivariate analysis. So we treated the methane in Spain or the methane in the Netherlands as a different trait and run it together. We have accounted for herd year season effect, robot, lactation week, and parity. And just to tell you a little bit about the validation, how do we evaluate whether actually we gain something in the accuracy or not. So we divided the whole data set into a reference and a validation population, where we did a forward prediction, which means that the youngest cows in each populations were used as a validation set. You can see this here on the right side, and all the rest of the animals were used in the reference set. Given that the Polish data was quite small, and we did do first analysis, however, it turned out that it didn't change any results. So I'm presenting here only the results when we combined data between Spain and the Netherlands. Looking at the results, so here you can see the genetic correlations for methane concentrations between the three traits. So looking at the results, so between Spain and the Netherlands, a correlation of 0.6 is really not too bad and very promising. So to use the data information across countries as an auxiliary trait could be really beneficial. The similar correlation of 0.57 between Netherlands and Poland, and between Spain and Poland, the correlation was 0.53. Good. Looking now at the accuracies of genomic breeding values with the three different scenarios. So just to remind you, we have assumed a zero correlation, then the actual estimated genetic correlation of 0.6 between Spain and the Netherlands, and a unity genetic correlation. Here the yellow bars shows the accuracy for Spain, and the blue bars the accuracy for the animals in the Netherlands. And what we can see, well, given that there was a much larger data set available for the Netherlands, so in general the accuracies were a little bit higher than for Spain. What we could see regarding increase when joining forces across countries, we could see that Spain gained more than the Netherlands when combining data across countries. So we could see an increase going from zero correlations to a unity correlation. And this is basically the actual scenarios with the actual scenario. However, for the Netherlands, given that there is already quite a large data set available in our reference populations, the gains were really minimal at the moment. So, well, basically that's what I just said. Take home messages from this part of the analysis. So Spain gains greater benefit than the Netherlands from sharing phenotypic data across countries, and including Poland with a limited data set at the moment did not yield in any additional benefits. Now coming to the second part of my talk, whether we could use the rumen microbiome in breeding for lower methane emissions. And I would like to acknowledge here David Flosters, but also Oscar, the two of them. They have been working closely together here and looking how we could use rumen microbiome data across countries for selection for lower methane emissions. So, but in general, how could we use the microbiome in breeding? So the first one could be, okay, we use it to predict a methane phenotype, and there are several studies showing that this is possible with a certain accuracy as well. However, at the moment, we are focusing on a very interesting method as well to use the rumen microbiome as a really correlated trait to methane emission. So what was available? Data availability was so we had data from Australia, from Spain and the Netherlands, and you can see the number of cows with rumen sample available. However, I have to mention from Australia, it was the oral microbiome collected via buccal swabs, but in Spain and the Netherlands, all the data was from rumen fluid samples. For all those animals, methane emission was available as well, where from Spain and the Netherlands, methane was recorded with sniffers, and in Australia, it was with grain feed. And then all samples were sequenced with long read sequencing. So first we looked at, well, just a comparison between the microbiome across the different countries. You can see to the very left-hand side, that's Australia, the oral microbiome, and then the Netherlands and Spain. And one thing you can see immediately is that the composition between oral and rumen is really different, which may have implications when we think of using the oral microbiome as a predictor of the rumen and ultimately of methane emission. Well, so something to take into account here. But very nicely to see that the composition between the Netherlands and Spain, even as different production systems, was really similar and gave hope so that we could combine the data here as well. So as seen before, so this is what we usually how to aggregate all those different taxa information from the rumen microbiome. So we did a PCA analysis, and you can see here on the right-hand side so nicely how the countries separate. So the green dots here, so that's Spain, and the blue dots here, so these are the animals from the Netherlands. And then just on a phenotypic level, what we try to do is to see if there is a connection to methane emission or already seen at, well, a phenotypic level. And you can nicely see it here for Spain. So where there is a shift, or where the animals with a higher methane average methane emission are located more here to the right. In the Netherlands, we did not see this to the same extent as in the Netherlands, as in Spain, sorry. However, this already gave some indication that the microbiome composition plays a role here for methane emission. What we did then as a first analysis, so we created an aggregated phenotype by PCA analysis on the whole microbiome information, estimated heritabilities, and then also estimated the genetic correlation between the PCA and the methane phenotype. So on the first row here, you can see the heritabilities for the different PCAs. Here in Spain, and here on the right-hand side, Spain, and here Netherlands, sorry. And you can nicely see so that there is genetic variation available for the different PCAs, which is already a very good prerequisite to use it in breeding programs. And then when looking at the genetic correlation with methane, you can also see that some of the PCAs are really quite high correlated, and some to a lower extent. Of course, here we have to mention that the standard errors are still very large. However, it is really a small data set. This needs to be taken into account. However, yeah, first results here look really very promising that we can use the microbiome directly as a correlated trait to methane emission in cattle breeding. In this regard, I would like to mention the initiative, the MicroHub project, which is a project within the Global Methane Genetics Initiative, which is coordinated in Wageningen, and the MicroHub is coordinated by Oscar from the University of Edinburgh. It's a collaboration between core countries, Australia, New Zealand, Denmark, the Netherlands, and UK. where we try to establish a reference population with metagenomic and genomic data. And the aim is to bring data together from different countries and use also data that will be generated within the Global Methane Genetics Initiative, where the aim is
Birgit, we lost you.
And so our first results show that the microbiome composition is heritable and genetically correlated to methane emission, and that the microbiome could be really used as a correlated trait in breeding programs, which would be really cool result. And with that, I would like to thank you for your attention and listening.
Thanks a lot, Birgit. I think there was a few issues, so we missed a sentence from the last bit, but I think it was very clear, though, your presentation on how to integrate genomics for lower emission in breeding. But so now we'll move on, but we'll stay with livestock for our final talk. If I'm not wrong, it's a shift from ruminants to pigs. So our next speaker is Luca Fontanesi from the University of Bologna. He also is the chair for EU Life and works in livestock. But today he will show us how metabolomics can help us link genetics to metabolism and resiliency, and how this insight can support more sustainable breeding. So Luca, I'll give you the floor.
Okay, thank you. Thank you very much, Gina. And I would like to thank EFAB for organizing this interesting webinar series, and Ecogen, of course, for inviting me. Birgit already explained a little bit what in real livestock we are doing. For the pig, we are dealing with the other aspect of resilience that is related to the adaptation to stressing conditions. In the work package in which we are working, the breeding work package, we are collaborating with the Italian Pig Breeders Association, ANAS, PIC, and CSIC, and Poznan University of Life Science. So we'll report a few elements derived from this collaboration. And of course, first of all, I would like to mention all the collaborators that did a great job to produce the data that we are publishing, and I will talk about some of the data that we have produced, in particular from the Animal Food Genomics Group, my group, Samuele, Matteo, Francesca, Giuseppina, Anisa, Valeria, Paolo, Maria Letizia, Stefania, and the collaborators from PIC, Maurizio Gallo from ANAS, Craig, Justin, Bruno, PIC, Eva from Poznan University Life Science, and Maria Munoz from CSIC. Just a few words about the metabolites. Probably we don't need to introduce everything, but just to focus on a few elements. Metabolites are end products of cellular process, act as intermediate phenotypes. We call them molecular phenotypes, and they are proxy for final phenotypes. They can explain some elements of the final phenotypes. So with metabolomics, we want to characterize all these moleculars, low molecular weight metabolites in biological samples. Here there are a few issues, of course, because depending on the time in which we collect information, we collect the samples, the way in which we analyze the metabolome, and so on. Metabolomics is in the middle of the technologies that want to identify internal phenotypes and molecular phenotypes. They produce metabotypes that are the level of the metabolites in biological fluids, usually in a tissue, sometimes. And of course, in this context, it is important to relate the level of the metabolites, the metabotypes with the genome level, environmental factors that can modify the level of the metabolites and related them to the production traits. The external and final phenotypes that are the economically relevant phenotypes. We are actually dealing with genetically influenced metabolites. So we want to identify the metabolites that are genetically influenced because, of course, we want to apply them in breeding programs. So we call them GIM. So we developed a few experimental designs based on a few pig populations. We divided the pig population in which we worked with in large cohorts and small cohorts. In the large cohorts, we can produce information by combining metabolomics and other information, genetic information. We can obtain information to explain biochemistry, the physiology of the animals, the genetics and the genomics aspect of the level of metabolomics, of metabolites. In the small cohorts, we don't have the power of explaining genetic factors, but we have the power to explain physiological response to some challenges. This is the rationale in which we worked. So we work with large cohorts and small cohorts. In both cohorts, we analyzed more or less the same metabolites, and we have many metabolites that are, of course, in common on the two designs. But of course, we are interested only in a fraction of these metabolites, the GIM, the genetically influenced metabolites. And in the small cohorts, we are identifying metabolites that are involved in the response to challenges. Some of them can be a part of the GIM. So this is what we are interested in. Why this nested design? Because it is always very difficult to challenge the animals in large cohorts. So it is almost impossible sometimes, and it would be too much expensive and not possible technically. The large cohorts that we work with come from PIC, Duro pig lines with a feed intake, stream animals for feed intake. From ANAS, we have two populations, Italian Duroc, Italian large white. On the wall, we have more than 1,400 animals. We collected blood for these animals, and we produced, of course, genomic data on these animals. The small cohorts, there are several small cohorts populations, local pig breeds, Mora Romagnola, Citta Senese, Italian large white, and Italian Duroc, the two cosmopolitan Italian breeds, for which we tested the effect of heat stress, actually tested in a circadian rhythms, because we collected the samples in the very hot summer period, late in the afternoon, the hottest part of the day, and early in the morning, the coolest part of the day. And we compared this information. The same experimental, most similar experimental design was done by CSIC for two populations, Iberian and Duroc per Iberian, collecting samples in 3 time points. Similar experimental design was done by Poznan University of Life Science with 2 local pig breeds, Slonicta white, Slonicta white per Slonicta spotted, again with 2 time point collection of the samples for metabolomic data. Metabolomic data been obtained using 2 platforms, Untargeted Metabolomic Platform and the Target Metabolomic Platform on plasma. And we have more than 1,000 metabolites divided in many different super pathways covering lipid, amino acid, and xenobiotics, nucleotides, and so on. So go back to what we can extract from these data sets. We can go to the biochemistry and the physiology using larger cohorts, and the physiology using the small cohort experimental designs. Only metabolomic data Just an overview of the physiological mechanism, an example. And I use the example of the PIC. Here is just the raw person correlation between all metabolites of various super pathway. So super metabolites of the super pathway are indicated with different colors, and here are the correlation. Okay, so there are many different type of information. We need to go into many details to extract relevant data. And of course, just a quick overview of what we can extract. We divided the pig from PIC in high feed intake and low feed intake, and we extracted, we subtracted the correlation between this group of pigs with this group of pigs. And the result is this one. And the result is this one. What does it mean, the result? This is the level of the person correlation. And in the lipid, we have a higher correlation with animals with low feed intake. Higher correlation means not the level of lipids, but a strength in the metabolism of the lipids. And here we for the nucleotides we have higher correlation in the high feed intake pigs. That means there is a higher level of metabolism in this class of metabolites. Quite interesting to have a quick overview of what can be extracted from this type of data. From the biochemical point of view, we used the Gaussian graphical models using partial correlation coefficient that are pairwise person correlation coefficient conditioned against the correlation with all other metabolites. And here is just an overview of the result. Of course, I cannot go into many details. There is the possibility to reconstruct the biochemistry of the metabolites. So we can reconstruct many pathways of metabolites, novel pathway, confirming pathway that have been already known, just using the level of the metabolites in the plasma. In the pig population constituted by small cohorts, we can identify the group of metabolites that can explain the true difference between the two time points, late afternoon and early morning. This is just a quick overview of the result obtained. Using the whole profile, metabolite profile, is not possible to identify, to differentiate the two time points, but selecting some of the metabolites with some specific methodologies, we are able to identify the metabolites that can really differentiate the two time points and the physiological state of the animals here. If you want to just go to the genetics and genomics aspects, we need to, of course, use the large cohort, and we can just analyze the level of heritability, because this is quite important, the GIM, and this is what we obtained using our thousands of metabolites. This is the level of heritability of the different classes of metabolites, and in the three different populations. There is a quite interesting heterogeneity between populations, and a very interesting distribution of the level of heritability that goes almost from 1 to 0. Of course, we are interested in metabolites that are genetically explained in some way. If we go to the other level, we can include also other genomic information. The GWAS, here is just a quick overview of what we can obtain. That is just a joint Manhattan plot summarizing the result of the GWAS using the UnTarget platform. We have obtained many different peaks, and for most of these peaks, we identified the right gene that is involved in the specific pathway that transform one amino acid into the other one, or one lipid into the other one. So just using using a few hundred animals. And this is an overview of the GWAS and the other two large populations, the Italian large white and the Italian Duroc population. And this is what we obtained using a single metabolites, and here what we obtained using the ratio between the metabolites, because we can also multiply the type of information, phenotypic information, using the ratio. And meta-analyses are quite informative, and it is interesting in any way to see that the two breeds are quite different in terms of genetic factors affecting the level of many metabolites. Again, here, what we obtained is merging the two type of experimental design, this fraction of metabolites that are very interesting for breeding purposes. In conclusion, metabolomics contributes to overcome the phenotypic bottleneck because we can produce tons of novel molecular phenotypes, including also the ratio of the metabolites. We can explain peak metabolism and resilience by combining results from different experimental designs. And of course, most of the results are also hypothesis generating, because we can identify something that was not expected, and we are dealing also with other approach to confirm and to take advantage from the tons of information that we obtained. We have published a few papers recently, and if you are interested, you can take a look at many other results that we produced. And I would like to recognize again Livestock, ULife, and also the partners in the study, ANAS, PIC, CSIC, Poznan University of Life Science, and all the collaborators that did a great job, and you for your attention. Thank you very much.
Thank you very much, Luca. It was a really interesting and straightforward presentation, and really it attached itself to the different presentation we had today. This concludes our speaker presentation for today, and we've heard from 5 excellent examples from epigenetics to cell models to HoloRuminant, genomics, and metabolomics. So each showing a different way to how to move from beyond traditional breeding. So now I'd like to open the floor for questions and discussion. I invite the speakers to switch on the camera so we can actually discuss. And if there's any question from the audience, I would be really happy to have you. If you're shy, you can drop a question in the chat box. So if there is no questions, I think I will maybe start it so we can have a nice chat. So we've seen a lot of different advanced tools today, and I think from epichips to HoloRuminant to metabolomics, I would like to ask each of you, because we are IFAB and we represent breeders and we have also researchers, but how do you see this approach, how those approaches will reach farmers and breeding organization, who it will reach first, and how can we make that happen or help it happen? So I would like to hear from you.
Maybe I start. Well, we have close collaboration here with the breeding organizations already, actually. So they are partners in the project. They are providing the relevant data that is needed for all our analysis. So specifically now for the livestock part, actually, so this information has already contributed as well to the implementation of a breeding value for methane emission in the Netherlands. So the whole genomics part, I think that's already quite nicely connected to the industry. With this also links to the farmers, because ultimately the farmer is the one using the breeding value or using the tool that we are developing here. So for the more newer part, I would say, so that's more when it comes to how to use the rumen microbiome. So that's still a little bit on the research phase, I think. So there it's just that we need more resources to really make this applicable really for routine analysis. You know, for animal breeding, we always need a lot of data to estimate breeding values accurately. So it kind of, yeah, and rumen sampling is quite an invasive technique. Of course, we know that, however, at the moment, it gives the most accurate information that we need for microbiome composition. So there's still a little bit of.
Okay, if I can jump in, because we got to the microbiome at the end, and I was actually happy to see that you got so promising results for the microbiome for use potentially even in breeding, because I've seen different results that people get for the heritability of the microbiome in general. I think it's still a bit unclear where we are on that, and still we are mostly looking at bacteria and archaea, so it's very, the protozoa, the other eukaryotes are usually not even included, and they are a big part of the rumen community. But as for how the tools that I use in general could be applied and sort of brought to the downstream use, I think I liked what was in the last presentation by Luca, saying that this hypothesis generating research that we do, a lot of what I do also falls in this category. So we are coming up with really interesting leads that could be super important, but it takes a lot of development further on to actually sort of confirm the hints of relationships and mechanisms that are interesting. So there is not such a straight line from my work to having something applied. But I mean, all important breakthroughs start in basic science. So that is what we have to do. And even if it's not clear what the endpoint will be, it is so important that we do this work. And that's where all the good ideas are coming from then.
Thank you very much. We have a question from the audience. Paolo, I give you the floor.
Yeah, yes. Well, my question is, I have two questions. The first one is, I pose one problem. The two problems I think I would like to have your opinion on. One is the validation of what each experiment is producing in terms of either metabolites, epigenetics, or whatever. And the second is how can, for the application to breeding, how can these results can be scaled up? Because then if you go in the field, then you need to have a cheap and feasible assay to measure these phenotypes.
Maybe I can try to answer. Thank you, Paolo, for this question. Actually, for validation, we are dealing with different platforms. We are using different platforms, for example, target and target metabolites, metabolomic platforms. Some of them are partially overlapping. So we obtained the correlation of the level of metabolites around 90% between platforms. So yeah, I think it is a good validation considering the platform work in very different way. And we are also working with different populations. We obtain more or less the same result, applying the different platforms in different populations. So this is by definition in some way a validation. And of course, another level of validation is, okay, we identify, for example, a metabolite affecting the level of an amino acid. And if we go in some way in a GWAS, we identify the gene that is involved in the metabolomy, the degradation of the transformation of this specific amino acid. So again, is another level of validation. And for the implementation, we don't want to implement to analyze thousands of animals in a breeding program. We want just to analyze the nuclei, of course, because based on that we can in some way transfer the information to many other animals. So our idea is not just to prepare new phenotypes for large scale use in the field. Maybe we can reach also this possibility, but our aim is just to analyze the pigs in the nuclei that in some way can transfer the progress to the based to the basal level of the production structure in pig breeding in some way. And then we need to we want to use metabolomics to identify novel traits that cannot be explained or maybe are difficult to be explained using traditional methods. So this is what we want to do.
Thank you very much, Luca. Terhi, I have a question for you. You talk a lot about the cell models and exploring the genetic resistance in cattle. So because Birgit mentioned before that there is a lot of invasive methods, and you've mentioned that cell models can replace those invasive trials. So how do you think this can really change animal health research? But also a second question that goes with it, because you worked on mastitis. So do you think that this approach that you use could be expanded to other disease or stress conditions?
Yes, thank you. Yes, so to answer first to the last question, this is really like easily to expand to the other traits as well, or diseases as well, because it doesn't really matter what are the pathogens or substrate to use with the cell models. And the first question you said, you asked about the how to, can you repeat that? Sorry.
You said that those cell models can help like reduce the invasive methods that we can use. And how do you think this can be transformative for animal health research? Because there's different work, like P-Web, who's also working on non-invasive methods. So how do you think this can be an innovation in terms of research also? Because we do research for food, right?
Yes. And there are some possibilities to, of course, to use it and really to improve the animal health, because what I especially like there is that they are what we do there is the non-invasive approach, totally non-invasive. Cows are just milked as they would do as normally as well. But of course, there is limitations because it's it's not reflecting the whole picture of the, for example, so let's say that we are able to get valuable data there, but it's not totally avoiding the use of true animal models. So.
Thank you, Terhi. I think it's still interesting, and I think that talking about like how you realize all the speakers, there's so much connection that could be used. So how, like Adrián, everyone, how do you see like what you heard today? Which type of data that you heard and you said, okay, this could actually be super interesting for like to add to my research and to further it. So is there? I mean, I could already see Velma, Birgit, and Luca looking at each other, and that there's so much interesting things. But Adrián, for example, like, do you see that epigenetics can be included in the research that's being done in livestock and in HoloRuminant? So how do you see the future?
Absolutely, but we will get crazy if we have to add epigenetics to microbiome research, because yesterday I was in a talk and they started to do epigenetic research on bacterial strains. And if we do not know anything, and don't quote me on this, about mammalian epigenetics, I think we are even more lost in microbiome epigenetics. It's like, I think we're in a stage where we have a lot of sources of information. Maybe we have too many sources of information, and I totally agree with what Velma said before, that basic biology, I think it's really important, and we need to be really, really sure of what we are finding before we are able to go to the farmers, to the breeding organization, and say, hey, I have this idea, we have these findings, and we would like to apply them to the actual systems. And I think that's our biggest responsibility. We need to be very, very sure about the validation of the results. What Paolo was mentioning in our side, we're doing quite a lot of things with that. We are using all the current methods and see the correlation between them. But yeah, I think all these European funded projects benefit really much from each other, but we need to do, and coming back to the first question you asked, we need to really be sure what we are doing and the result we are getting, because they are really cool from a basic biology point of view, but we need to have this responsibility of be sure how we transfer them to the actual system.
Thank you, Adrián. Luca, maybe you can add on it, and maybe it would be interesting to know how cost action like EU Life can help in those transfers.
Yeah, thank you. I want just to jump in to confirm that what we want to obtain is something that is out of the box in some way. Adding additional level of information, we can discover something new, something that was not possible to obtain or to even imagine using traditional tools. For example, using metabolomics, we are understanding better how the metabolism is running in the pigs, and we can even modify the diets according to the level of some of the information that we obtained based on the genotype of the animals. So we can go in a very unexpected precision feeding approach, for example, based on the genotype of the animals, but based on the genotype of the animals that are related to the nutrients that in some way are obtained, are transformed by the animals, for example. And for the cost action European Network on Livestock Phenomics, of course, phenotyping is the new frontiers. Of course, we have the genomic tools now, but we need to, in some way, feed the genomic information with additional nutrients in some way, just to stay on the field. But the nutrients of the genomic tools, the new nutrients are the new phenotypes. Because if we don't have new phenotypes, we cannot understand the very basic biological mechanism that in some way can reach the final level of the economically relevant traits that in some way are really difficult to be improved. For example, disease resistance, behavior, welfare, and other very difficult traits that we use in traditional methods are difficult to be even understood.
Thank you, Luca. I think that's exactly what we need, right, from collaboration like this. I would like to start closing this webinar today by asking you, each speakers today, maybe to give one practical outcome from your work, maybe effectiveness of application, or what do you hope from your research to bring to other researchers, or breeders or farmers, but also policymakers, because there's so much done at the cellular level that needs understanding for better policy decision. So should we take away from today's discussion? Maybe we start in order, Terhi.
So actually what we found out are the biomarkers that can be used for the breeding programs to implement how to calculate in the genomic breeding value calculations. So that's what the cell models can offer for us, I think, at the moment. So they are increasing the basic knowledge of the infections in this case, and then that can be implemented in the breeding programs.
Thank you, Terhi. Velma?
Well, I mean, I'm just going to reiterate the point that I already made, that we need to think about the microbes as well, because essentially, I mean, microbes are where the methane comes from. That is an issue that we want to mitigate, and microbes are what is digesting the feed. So we really have to think about microbes. And specifically from my research, as a message more to other researchers, is that I hope we go back to looking more at protozoa, because they have been not so much studies, and we are finally getting to the point where we are actually able to look at them, partly because there are better reference genomes coming out. So this is, I think, a part of rumen microbiology that is going to be moving ahead in the very near future again.
Thank you, Velma. Adrián?
I'm very excited about the use of this epitip, and within the project, every group is using it for different traits, for studying different traits or different equations, and see how we can make this transfer of the information we are getting from this European project to the actual farmers. I think that's the key to have to find this point of collaboration where we validate the results all together, but with the idea of that we need to get back to the farms.
Thank you, Adrián. Birgit?
I would say, well, for us, the most important message is basically to the whole sector that animal breeding is a very important tool for to help climate change mitigation, specifically for methane. So there is a lot of pressure on farmers, as you can, for example, see in Denmark, they have implementing a tax for farmers. So it's really the goal for us to really show that there is a genetic background and that genetics actually can be part of the solution. And that's really, for policymakers, but also this message needs to go through the whole sector. And I fully agree with Velma. So I'm very excited about the microbiome, actually. So.
Thank you, Birgit. Luca?
Yeah, well, animal breeding is the engine of the animal production sector. This is clear, and is the green engine because it is cost-effective, cumulative, and is permanent. We need to innovate this engine using new technologies, and to do that, there is the need to collaborate between projects, groups, and European Network of Livestock Economics. The cost action can provide a good environment to connect people. This is, the future is bright if there are funding resources in the system. Otherwise, the future will not be very bright.
That's true. Thank you. Thank you very much, Luca. Thank you all for all those great thoughts. And I think your work is really inspiring, I think, for all the scientists in the room today, maybe from young and for more established scientists. I think it's great to hear how those projects are helping in moving animal breeding into a more integrated and sustainable way. And as all of you mentioned, directly or indirectly, breeding and genetics is part of the solution, no matter at what level. So on behalf of Ecogen team, I'd like to thank you all, the speakers, but everyone who joined today and who stayed, even if we're like 10 minutes running out of the actual time. We hope that you will continue to follow the Ecogen series. The next series will be in November, and we will discuss still beyond genetics, but we'll focus more on gene editing. And thank you once again, and I wish you have a great day and the rest of the week. Thank you.